So, what was the major difference that we found when it comes to exciting a smooth muscle versus an versus what versus a skeletal muscle? The skeletal muscle is simple and straight. What is the agent that we need to use? Skeletal muscle can be excited with what? You are still. I need to wake you up with acetylcholine. Okay, acetylcholine coming over what fiber? Coming over the motor fiber from spinal cord, whatever. Okay, are you with me so far? In the case of smooth muscle, however, it's very complicated. There are n number of agents. Okay, right from stretch to chemicals to and the thing is there is enormous variety in which a smooth muscle can be excited, whereas a skeletal muscle can be excited but cannot be inhibited. The only way to inhibit it is to let the acetylcholine be chewed away by the acetylcholine esterase, and then there is no acetylcholine and the muscle will relax. But here in the case of smooth muscle, you can have an agent that will excite, you can have a muscle that will inhibit, you will have a muscle that can relax. Both systems are extremely elaborate. What is further exciting is you cannot have, listen to this, you cannot have the excitation of a skeletal muscle without action potential. Hello, action potential has to be there. Okay, there has to be the typical spike has to be there. In the case of smooth muscle, don't worry about it. Maybe, may not be, depends on individual source by which a muscle cell is activated. So, can a smooth, listen to this, can a muscle cell be activated without action potential? Yes, it can be. It can be, there can be. So, there comes an agent from outside, it excites a particular receptor in the plasma membrane, it takes a signal inside and finally, the signal goes to there is increase in calcium ion channels and the action myosin filaments slide on one another and the muscle contracts. At no stage did you encounter any, you did not encounter any action potential. Wonderful phenomena, it can do without action potential. Okay, but that does not mean that no, it can do, it can, there can be action potential, there may not be action potential, there may. So okay, so we have seen this. So, the calcium was the key element. On excitation, the calcium ions will go in, and then we have seen that the calcium will combine with this very interesting molecule called as what? Okay, calmodulin. Okay, this where, so it's not there, it was troponin. Okay, okay, here it is calmodulin. There, can you see 4 tiny red dots there? Each dot represents a calcium ion. Okay, and then when the calcium ion combines with it, this calmodulin molecule undergoes a configurational change. Okay, and then as a result of that, it combines with the it activates the kinase enzyme, what you call it as the MLCK kinase, and then it brings about the phosphorylation of this essential light chain, and that finally leads to the what? On the myosin molecule, there is ATPase that is inactive, that is activated, and once that is activated, it will start cycling, and you will have the contraction of the. So, this is just to give some more details of the same, and we have different look at a smooth muscle here. Okay, so far there is a plasma membrane. Okay, so far, yeah, there are different ways by which there could be a signaling molecule could come through a capillary, or it could come as a neurotransmitter released by a terminal, okay, either a hormone or a neurotransmitter, or it simply stretching it. So many things can give rise to, either it can combine with this G protein coupled system, this is the G protein, it activates what this is phospholipase C, it converts PIP2 into inositol phosphate 3, the inositol phosphate 3 goes and binds to this particular calcium channel which is sitting on the sarcoplasmic reticulum, and the sarcoplasmic reticulum releases calcium. So, you have plenty of calcium, it depends on what type of cell you are, maybe this, maybe this, maybe this, you have lot of calcium, rest of the story same, calcium combines with what? Calmodulin, and the rest of is inactive form, active form. This is, of course, I am sure you can identify, this is the cross bridge. Hello, cross bridge. On the cross bridge there is inactive ATP, it will become active ATP. Once it becomes active, then it will bind, myosin head will bind with the actin filament, and the sliding phenomena will happen. This is just for, this is sequence of events what we have seen just for this will help you to remember the steps. Now, whereas in this we saw how the what is the starting point? There is a signaling agent. which is coming from outside, okay, and that excites and you take a signal inside, cascade of events, finally you get a contraction of the muscle. Good. 2 points. Most of this action, this action is happening at the level of caveoli. Get my point? What is a caveoli? What is a caveoli? If there is it's a spindle shaped cell, hello, fat in the middle, okay, tapering at the end. What do you have in front of you? A smooth muscle cell. Okay, so far. The length is about 250 to 300 microns. The thickness may be about 2, 3, 5, 6, 7, 8, 9, 10, 15 microns depending on what tissue you are. And if you walk along the surface, you go and suddenly there is a pit. Go along, again a pit, again a pit. What is the technical name for the pit? Caveoli. It is in this caveoli, okay, it is in this caveoli you will find all those receptors are sitting. Where are they? They are in the caveoli, okay. And the second anatomical beauty of the caveoli is that from the caveoli there is plasma membrane which is like a pit. If you cross the plasma membrane and go inside the cell, you will inevitably find sarcoplasmic reticulum there anatomically. So, there is plasma membrane, occasionally there is a pit, caveoli, and immediately inside you will get what? Sarcoplasmic reticulum. So, this is we are already in a caveoli, this, and then you will have the sequence of events, and here we have a diagram that tells us as to how the contraction will happen. Does it mean that always contraction? No, we have seen, the author just gives us another example where can we see there are 2 circles? The 2 circles are you take a section right in the neck, okay, and in the front you will have trachea, and behind that you will have esophagus. Are you okay? Okay, in the pharynx, the nasal passage will go from your nose, okay, it will go, okay, and then you will have the nasal passage is going to the trachea, which comes in front, and the 2 passages cross. I do not have to tell you that anatomy. And then we have the esophagus and the trachea there. In the trachea, the author is drawing our attention to this muscle called as, what can you read it as? Trachealis muscle. It is a smooth muscle, okay, which is lining a part of the trachea. Are you okay so far? Okay. Now, you have an allergic reaction or something that you have inhaled that does not agree with you and the muscle has gone into spasm, okay, and then you want to relax it. How would your biological system react to that? Well, the sympathetic nervous system may be stimulated, the blood may release adrenal medulla may release epinephrine, sorry, it will release what? Epinephrine, okay, from the adrenal medulla, it will get into the blood, it will go everywhere. Along with that, it will go on the smooth muscles, and on the smooth muscle plasma membrane you have a receptor. What receptor is there? Beta 2 adrenergic receptor. It is a G protein coupled, and as you can see the protein goes up and down 7 times. We are okay so far? So, this somewhere here there is affinity for epinephrine. Okay, it will give rise to adenyl cyclase, ATP, cyclic AMP, and then finally it will downstream effect, series of steps, and then you will have the relaxation of the smooth muscle. So, what do you have? Relaxation. The muscle will relax. We have already seen. Okay, what was the first line of treatments? Acute treatment for the patient of asthma. Injection of what? Injection of epinephrine. Injection of what? In extreme cases, but that's used. So how does it act? So you have a patient in front of you, he is in a severe asthmatic attack, and the doctor gives him a shot of what? Epinephrine, adrenaline, and then you can look at this diagram and predict what is happening. It enters into the blood, goes where? Everywhere, and then it will specifically bind where? Smooth muscle. What receptor? It's there. Beta 2 receptor. I mean, I can't make it simpler. I mean actually I'm as if I'm talking to, or you think it is too simple and so it can be ignored. Okay? No, it's not important. It's not. So, okay, then you have entire cascade of events, and what is the net result do you have? Relaxation smooth muscle. So have a firm idea, okay, that the smooth muscles can be excited, they can be inhibited depending on, and actually we have been repeatedly addressing this point. Okay. Now, this is a very interesting anatomical peculiarity, anatomical physiological peculiarity of smooth muscle. You will enjoy this, just be with me. If I look at this, I know I do not have to tell you what is this. Skeletal muscle, 2 Z membranes, hello, anchored on them are the actin filaments, and then myosin filaments. We have done it 100 times. Good so far? And somewhere here is the M line, but don't. I want you to focus just here, just here. And supposing you are on the myosin filament. Where are you? You are sitting on a myosin filament. Okay? You are sitting on a myosin filament and you are facing the Z membrane. Okay with me so far? And then you have two oars in your hand. So that you are pulling the myosin filament towards the Z membrane, okay or not? Both hands, okay. You are pulling in the same direction. I am underlining the word. What the word I am underlining? Same word. Okay. So there is a cross bridge. There is a cross bridge on this actin. There is a cross bridge on this actin. There may be on one more actin. Let us not bother about it. But you are all working in the same direction. Are you okay with me? Are you okay so far? Now, let us change the focus to the smooth muscle, and I will draw your attention to the let us say this at the top. Okay, can you see a dense body there at this end? Okay. Can you see the dense body there? Can you see the dense body there? Yes or no? In the anchored on the dense body is the red. What is that red? It is the actin filament. Okay so far? And then this is that green thing. That green thing codifies myosin, and then another actin, another myosin, and the last one is anchored on the dense body. Okay so far? So how many myosin filaments can you count there? 3. How many actin filaments can you count there? 4. Okay so far? Now listen to, look at this myosin filament. Whereas that myosin filament, whereas it pulls this actin filament in this direction, it pulls the other myosin in the opposite direction. Hello, are you with me? And then look at this one, look at the middle guy, look at this middle myosin, in which direction it is pulling this actin and in what direction it is pulling that actin, same or different? Opposite direction. And that is yes or no? Yes. Then that is very well shown here. The author simplifies that, supposing this is one dense body, this is another dense body acting and anchored on them are acting filament, this is the myosin filament, and this myosin filament has a crossbed, pulls this actin filament, look at the arrow, this direction, and this actin filament in the opposite direction, a phenomena that can never ever be seen in the case of skeletal muscles. An amazing phenomena, okay, opposite direction. As a result of that, whereas a skeletal muscle can contract from whatever it is to 70%, are you with me? Whatever is the distance between Z membrane, and when it contracts it can bring down to 70, 90, 80, 70%. Because of this interesting arrangement, from 100 it can go to 30, 100 to 30. Are you with me? Yes or no? It can pull so much because of this very interesting arrangement of the actin myosin arrangement and the cross bridging and the whole. We are all on the same page, everybody? Okay. Just appreciate the beauty of the smooth muscle here. So just see, because there is a myosin filament, it will pull actin in this direction, and the whole cell can become very small. The two tips will be 30% as compared to the original.
Even here the polarity of the actin filament is separate. So the negative
side
of the actin filament.
Yeah. It is same. Okay. Then there is yet another trick up the sleeve of actin myosin filament. I talked to one of you, I don't remember who. When we studied the cycling of the actin myosin filament with reference to the skeletal muscle, there was a transient step which we call as rigor. Rigor, okay. And when you die, there is rigor mortis, and that happens because actin myosin filaments they remain attached to one another and the body becomes stiff. Okay, that is rigor mortis. But forget about it, just rigor. And it is a transient; it just comes and within a few milliseconds it goes. It is a transient step. Okay. Now this actin myosin filament remaining attached to one another for quite some time is a regular phenomena in the case of smooth muscle. We do not never ever use the word rigor. we use the word latch formation. What word do we use? Latch formation. Latch formation means, okay, what is the advantage of that? The advantage of that is, supposing I want to lift half a kg of weight, okay, as long as I am holding it like this, all the muscles of my hand, actin myosin filaments have to cycle and cycle and cycle and cycle as long as I am holding it. Okay, it's cycling has to happen, and for every cycling I need to use 1 ATP. Okay, so skeletal muscles are expensive. Okay. Now, in the case of smooth muscle, this amazing physiology has evolved where everything is slow, go slow, and when you want a particular muscle to remain contracted, okay, for 5 minutes, 10 minutes, 15 minutes, the food has entered into the stomach. The stomach is vigorously mixing the food with the enzymes and the acid. You do not want the food to reflex back into the esophagus. You do not want that to happen because the esophagus is not equipped to deal with the acidity, and the acidity may be pH 2 to 2.5, it is very acidic. Stomach can deal with it, but the esophagus can't. To ensure that it does not happen, you need to have a valve where at the junction between the esophagus and the stomach, and the valve needs to remain closed for 15 minutes, 20 minutes, 30 minutes. Okay. In such a scenario, the smooth muscles show the phenomena of latch formation. Means what? Let the actin myosin filament remain joined, let the muscle remain contracted, okay, for as long as it is being told by some signal that you remain contracted. Why you remain contracted? Because you do not want the food to go reflux from the stomach into the esophagus, and that is going to take half an hour. Okay. So, remain contracted, and during that period I am not going to use any ATP. How can I do that? I can, if I can somehow manage that the smooth muscles of the valve that guard the opening from the esophagus into the stomach, if I can make the muscle remain contracted for half an hour without using energy, perfect. That is exactly what the smooth muscles do. And let us see, and another beauty of this. The real beauty is we do not know how it really happens. Okay, we do not know. Okay, we have when we do not know, we hypothesize. Okay, we suggest. Okay, and there are a couple of theories floating around that try to explain as to why this is happening, and I will introduce you to one of them. Let us look at the okay. Now, I will now introduce you to two very basic words. I want you to understand and remember those two words. Let us look at the smooth muscle of the stomach. Okay. What about it? Well, you have taken food, it has gone from the esophagus into the stomach, and as a result of the arrival of the food, the stomach has released its acids, its enzymes. How that happens, I will tell you maybe in the next lecture or whatever. And then the wall of the stomach undertakes vigorous contraction relaxation activity. Actually, it starts as if this is stomach is a bag. I do not have to tell you, you have all seen the studied and drawn the stomach. Okay. From the rostral end, immediately after the vagus, a contraction wave starts, sort of peristalsis. It goes all the way to the duodenal end, which is called as pyloric end, and another wave comes and another wave comes. But the pyloric sphincter that guards the opening from the stomach into the duodenum is also closed. Why? Because the food is not ready to be discharged into the duodenum. It has not been digested. All the enzymes of the stomach which need to act on it have not yet acted. So, you need that 15, 20 to 30 minutes of time during which the stomach is on its own, and there is a wave of peristalsis, another wave of peristalsis, another wave of peristalsis. Every time a smooth muscle goes through a wave of contraction and relaxation, which may take 30 seconds, which may take 1 minute, which may take 90 seconds, I will call that as the phasic contraction. What do I call it as? Say that again. Phasic contraction. What do I call it as? Phasic contraction. It comes and goes. Okay. Now, why is it coming? Why is that happening? That is happening because the smooth muscles of the longitudinal and circular muscles, okay, alternately, okay, they contract, stay contracted, contract 30 seconds, 40 seconds, 60 seconds and then relax. But the next one will contract and the next one, this is how the wave will go from the anterior end to the caudal end. Are you okay so far? I am just describing how that peristalsis we have done it. Peristalsis is happening at the level of stomach we are focused on. Why is that muscle contracting? Why is that particular circular muscle contracting? Because it is being told by whom? By the mind trick plexus. By whom? Mind. How does that happen? That also I will tell you. By the mind trick plexus is doing it. So, what is mind trick plexus doing? Doing a stimulation. To whom? The smooth muscle. What smooth muscle? Circular smooth muscle, which it is there in the neighborhood. I am going to show the mind trick cell which is going to stimulate the smooth muscle. Okay, you contract, remain contracted for 30 seconds and then you relax and then the other guy will contract and the other guy, and this way the wave of contraction will go from one end to another end of the stomach, and a second wave and a third wave and a fourth wave. Are you okay so far, everybody? Great. This, if you look at a single muscle there, you will find it contracts and relaxes. I will call that phenomena as, again I am explaining, what do you call it as? So we are stimulating it here. How are we stimulating it? We are stimulating because there is a neuron of the mind trick which is releasing an excitatory neurotransmitter on the smooth muscle, and the smooth muscle is contracting in a phasic manner. So, it is stimulating, that stimulation stays for 30 seconds, for 30 seconds the muscle is contracted and then the stimulation goes away, okay. Then the next one and the next one, in this way the wave goes from one end to another end. Done so far? Okay. Now, whereas that is the story at the level of a smooth muscle which is lining the stomach proper, I am talking about the esophageal sphincter and the pyloric sphincter. The names are self evident. Esophageal is esophageal and pyloric is pyloric. These, on the other hand, once you have taken whatever food you have taken and then the esophageal sphincter, okay, now knows that it is time for me to contract and stay contracted. Why? Because it has because that sphincter of this has been told by whom? By again the mind trick plexus, where that mind trick plexus of this part is now telling the smooth muscle contract and stay contracted for 30 minutes. You will call the phenomena as what? Call it as what? Tonic contraction. So, have you fully absorbed the meaning of the two words which I gave you? What are the two words I gave you? Phasic contraction and tonic contraction. Okay. But look, both of them will need a stimulation. They have to be, and then let us see what happens during. So we are looking at a smooth muscle. That smooth muscle is somewhere in the wall of the stomach. Okay, that smooth muscle has been stimulated by an excitatory neurotransmitter from a neuron of the mind trick plexus for a very brief period. As a result of that, within that cell something has happened with reference to this peak. What is that peak? Read for me. The first one, calcium has gone up. We have seen that 100 times. Calcium has gone up, makes sense. Gone up, gone down. Spike in calcium. This is followed by the second one, the blue one, which is called as what? Read for me. Velocity of what? Cross bridge phosphorylation. Okay, it goes up and that also goes down. And this is followed by the red curve. What is the red curve? The force of contraction, where actually the actin myosin mechanism. We have seen in details all the 3 steps. They are there. Okay. Now, whereas this is happening for every 30 seconds. So, 30 seconds one wave has gone. After 2 minutes another wave. Phasic. Done so far? Now, author tells us about this sphincter muscle and this sphincter muscle, and in the case of these two sphincter muscle, the stimulus comes and stays for quite some time. Okay, so far? Now, as a result of the arrival of the stimulus, means what? A particular neuron of the mind trick plexus which has released an excitatory neurotransmitter on the smooth muscle of the sphincter, and in that smooth muscle again calcium has gone up, no problem. The velocity of cross bridge phosphorylation has gone up, But after that something interesting happens. You know that kinase activity was there. Kinase, without kinase nothing will happen. What was that ML key? What was kinase was there? You know, I have forgotten. Very good, very good. That kinase, okay. That kinase is there, which takes the muscle towards contraction, and there is another phosphatase is there, okay, which will end, which will terminate. The authors tell us, the hypothesis tells us that there is a delicate balance between the two, and both of them go down. Both means what and what? Phosphatase on one hand and activity of both in a balanced way goes down. How it happens, I do not know, magic, that happens. Okay. Therefore, I want you to read the bottom 3 lines. How does this happen? One hypothesis is that the balance between the activity levels of myosin light chain kinase and myosin phosphatase determines the rate at which cross bridges cycle. The activity of both is depressed. Okay. I am sure you are confused. So am I. Okay. But that only tells us what? What tells us what? This is an unsolved problem. Okay. How it actually happens. Okay. Okay. Good. Good. Go. So, but because of this phenomena, just see how far these two smooth muscles remain contracted. Is there cycling happening? There is no cycling happening. No. This is as a result of this, it has determined there is no cycling happening. Only the actin myosin filaments, they just remain in a what state? I gave you the word latch state. Say that again. In the latch state, they remain contracted in latch state without using the ATP. An amazing phenomena how the nature has found a solution, that when you want to keep a muscle contracted for a very long time and you do not want to spend energy on that, then engage a latch phenomena there. And
so the sphincter smooth muscle and the wall, stomach wall smooth muscle are different, or the mitral plexus which are terminating are different?
Shall I tell you something? All the answers could be true. Absolutely true. Okay. He is asking a very interesting question. He is saying that, first of all, I do not know the answer, but I am trying to make a guesswork. He is saying that, okay, let me take the sphincter muscle. Let me take a sphincter muscle and let me take a muscle from the wall of the stomach. Okay, take two pieces, extract myosin, and let me take a look at, is this myosin identical with this myosin? The answer to the question I do not know. It may be identical, it may not be identical. Number one, even if it is identical, it is quite possible that there is a different neurotransmitter that is regulating this, there is a different neurotransmitter that is regulating the circuit that is for final, they all could be different. So, the answer could be anywhere there. Got it? Okay. Are we okay so far, or this is mumbo jumbo? We are good? Yeah.
Small thing. So,
Sure.
for the clarity of the 20 to 30 minutes during which they are contracting, stimulus has to be there.
Stimulus has to be there. Stimulus has to be, yeah. If the stimulus goes, then it will go back to stimulus has to be there. You are right.